Hi, good afternoon. I'm Toshifumi Minamoto from Kobe University, and now I'm serve as a president of the eDNA Society in Japan. I would like to talk about the developing the eDNA manual in Japan. I would like to introduce the beginning of the eDNA, first eDNA study in Japan. Our work began with an unexpected observation while we are studying the koi herpes virus, KHB. We measured DNA in aquarium water and found that there are very high concentrations of fish DNA in the aquarium. And this prompted a simple but powerful question. Can this DNA be used for ecological surveys? That idea was put from Dr. Hiroki Yamanaka, now at Ryukoku University. And that question launched the first eDNA efforts in Japan. And later, we published Japan's first eDNA paper. We demonstrated the successful detection of fish eDNA in aquarium and natural river water. It was basically the same idea as metabarcoding even before we started using NGS. And using colony sequencing, we could identify several fish species DNA from river water. And the key message was very simple. A cup of water can list local fish composition, species composition. And since 2013, we have launched an environmental DNA project under the JST CREST program, led by Professor Kondo. This is the photo of Kondo, 13 years, very young. And this project focusing mainly on marine ecosystems, and the primary objective was to develop methods for estimating the population size and the biomass through quantitative eDNA analysis. And in parallel, we also initiated the development of eDNA metabarcoding techniques for fish communities. Here, I will briefly present this project result on quantification of eDNA. This study was conducted in the Maizuru Bay on the Sea of Japan coast. For jack mackerel, this species, eDNA-based estimate of 22 million individuals were comparable with echo sounder estimate of 16 million. So in short, estimate from eDNA was fairly close to echo sounder estimate. This, the CREST project was also produced another major milestone, the MiFish primers, which many of you are well familiar with. MiFish primer enabled efficient fish metabarcoding using a short 12S fragment. Some of the key validation tests were conducted at the Okinawa Churaumi Aquarium. In these aquarium tests, we successfully detected the eDNA of fish species kept there, covering 57 families and 120 genera and 168 species, corresponding to the 93% of the total species present. This successful development of this method led to a large-scale survey that I will show on the next slide. With this MiFish, powerful tool of MiFish in hand, we took on next challenge. In 2017, we planned a nationwide synchronized survey covering coastal areas across Japan, including remote islands. water samples and filtration were conducted at more than 500 sites with the participation of over 100 of researchers. However, for such a large-scale survey, a standardized manual was essential to ensure the data quality. So we assembled the materials into standard sampling and filtration kit shown in this figure. And also we developed illustrated manual with clear photographs. This made instructions clear so people in different teams and different regions could work in the same way. And this experience of developing this kind of field manual later contributed directly to the standardized manual produced by the eDNA Society. And using standardized methods, we have mapped the detection point of each fish species. Then the distribution of fish species can be very clearly seen. For example, in Japan, Pacific herring, this species in Japanese Nishin, is caught only in Hokkaido. And eDNA results clearly show Hokkaido is the northmost island, this island. And eDNA results show that this species was detected almost exclusively in Hokkaido. On the other hand, Japanese anchovy, Katakuchi-iwashi, is found all over Japan except Hokkaido. From the results of nationwide survey, we can draw more than 1,000 such species distribution maps. After that, in 2018, the eDNA Society was established mainly by the members who had worked together in the JST CREST project. The goal was to create a platform that could support the growth of eDNA science in Japan and beyond. And the mission of the society was first to develop eDNA science as a discipline, and second, to promote research, method standardization, and social implementation of the eDNA technology. Third, to construct the scheme to share and utilize biological information. And fourth, to connect science, government, industry, and education to develop eDNA technology as social infrastructure. Through these activities, the society aimed to support both scientific progress and the real-world application of eDNA methods. The activities of the eDNA Society include annual meetings. I believe that one important role of the society is to provide a place where members of various sectors can meet and discuss. And we also held an international conference. The first international conference was held in May of 2023. And now we are discussing about the next international meeting in Japan, maybe in 27 or 28, it's not decided yet. And one of the key activities of the eDNA Society is the standardization of the methods. And we published an experimental manual in Japanese and in English, mainly for domestic purposes. And parallel with the publication of the manual, we also hold seminars to disseminate its contents. And through these activities, the eDNA Society contributed to the improvement and leveling of eDNA analysis technology in Japan. And the manual explains both species-specific detection of eDNA and metabarcoding of eDNA, and covering all steps from sampling to bioinformatics, including sampling, filtration, extraction, PCR or qPCR, and NGS analysis. And by making each step clear, the manual helps ensure that the results are comparable. Every step in the manual is illustrated with clear photos and diagrams. And these visual examples show exactly how each action should be done, from how to hold the equipment to how the filter should look like after filtration. Because users can see the correct procedures, and there should be much less confusion or variation in the field and lab. This visual guidance also helps new team members learn the workflow quickly, even if they have little experience with field sampling or molecular biological experiments. And this helps many groups work in the same way, which is most important for making the result comparable. And the manual was updated whenever needed. And the Japanese version was first published in 2018, and revised editions were released in 2020 and 24 and 25. The latest English edition was published in June 25. And the standards need to change as science develops, so updating them helps everyone stay aligned while still allowing new ideas to grow. And standardization has brought several important benefits. And first, it has made data easy to compare across different regions and different teams. And it has also been used in government guidelines. And the standardization helped bring eDNA into governmental monitoring programs as well. And in addition, it has supported the development of ANEMONE and ANEMONE Global. And finally, it has made it possible for citizen science groups to join safely and reliably. These points show that the standardization helps both science and real-world practice. And the value of standardization becomes very clear when we compare different researches, because we all use the same protocol. Surveys done in the Pacific Ocean and the Sea of Japan, and in the rivers and even in lakes in other countries can be analyzed in the same way. And this allows the data from many teams and many environments to be directly compared. In short, the standardization makes any research in any region comparable. And standardized manual was also adopted in the governmental survey guidelines, ensuring the governmental monitoring follows consistent methods. And the Ministry of Environment in Japan published guidance aligned with our manual, expanding beyond fish to amphibians. The guidance helps make sure that everyone uses the same methods. This governmental survey, called the National Census on River and Dam Environment in Japan, has been running since 1990. And 109 river systems and more than 80 large dams across the countries was covered, and each river or dam is surveyed once every 5 years. So far, 6 rounds have been conducted, and the current round is the 7th. And the eDNA survey for fish species were introduced on a trial basis from 2020, and now manuals are being prepared for full-scale introduction of fish eDNA survey start from this year. And so eDNA is moving from the research stage to full use in this national monitoring program. The manual also brought ANEMONE, and in ANEMONE, surveys use the standard protocol provided by the eDNA Society, so the data are directly comparable across sites and teams. So far, the network has collected more than 1,000 sites and over 5,000 surveys. These numbers may already be higher now. And I'm sure some of you here today know the latest numbers better than I do. And this data set from a strong foundation for national and regional biodiversity assessment. And ANEMONE is now expanding ANEMONE Global, as you know, aiming to build an international framework using shared standards. This shows how standardized methods can connect local monitoring efforts into a broader global network. And the standardized methods allow citizens to collect eDNA safely and correctly. And simple sampling kit enable anyone to join surveys across Japan even without scientific training. And this public participation greatly expands sampling coverage and frequency. And because all samples follow the same protocol, citizen science data collected with the same protocol can potentially support national and global monitoring. So far, standardization in Japan has progressed quite well, but at the same time, we still face several important issues. And first, there should be a shortage of analytical companies to meet the rapidly increasing demand. And second, we need to ensure the quality of these companies, and particularly in terms of contamination control. And we need proper training systems and technical certification for the analysis companies. And finally, we must consider how to incorporate new technological advances after the manual has been published. These points show that we still need to keep improving. As eDNA use expands globally, many regions are building their own systems. This slide shows one US example, eDNA Atlas, and which is publicly available. Some of you may know the US situation better than I do, so I'll focus on the main message. As more databases appear, difference in methods and metadata become more visible. And that is why shared standard and international coordination are so important. Here is another example, this time from China. China operates an eDNA database called eDNA, which covers a wide range of taxa. And the main point is that many regions are now creating their own eDNA databases. And as these grow, difference in methods and metadata become more noticeable. And I believe metadata will be discussed in more detail later in this session. We can now see eDNA data from many countries and regions around the world. However, different places use different methods, such as filter pore size and eDNA extraction methods, and other primers and pipelines. And the coverage of genetic information in databases is also different across regions. For example, in Japan, more than 99% of freshwater fish can be identified by MiFish primers, MiFish analysis. But this level of coverage is not the same in the different regions. And the main challenge is how to make these data sets comparable. To achieve this, we need global standardization for eDNA analysis. In conclusion, the standardization of eDNA methods has created a reliable and comparable data foundation in Japan, and it has enabled a large-scale monitoring and contributed to the international networks such as ANEMONE Global. And the standardization is not the final goal. It is the basis for generating trustworthy biodiversity data into the future. And the final take-home message is this: let us move forward together to build a global future where eDNA empowers biodiversity conservation. I would like to thank the members of the eDNA Society and the students and the staff at Kobe University, and Kondo-san and members of ANEMONE team. Thank you very much for your attention.
Thank you so much for Minamoto-san. So we have a couple of minutes to take any questions and comments. So any comments and questions? So the Morrison first, and then come to Christian. It's okay. Then Wendy.
I just have a quick question. Do you have any way to track the uptake of your manual? Is there any way to see who has taken your manual and who's using it? Is there a way for you to see who has taken your manual and using it?
No.
Thank you.
Yeah. Thank you very much. I think very amazing. In terms of standardization, because I noticed the volume of collection is also standardized. How do you think the identification efficiency should be subjected to the volume? Right? So this is something I think very important, because I saw we always use a toolkit with that kind of a sample volume. Do you think that kind of volume will also have some relation with the number of species which may be identified?
Yeah, that's right. That's an important point. Yeah. And our manual says to take just 1 liter of water and 1 liter of water. But in many cases, especially for marine environment, the 1 liter is not enough, I think. Yeah. So the water volume to take is very important point, but it is not standardized yet. In Japan, we how to standardize, yeah.
So that's why I feel when we talk about, we always use, not only eDNA, but also microplastic research, always different protocol. We all talk about we want to standardize. So people will ask, what standard is standardized? Right? So something I think from ourselves, I think through the research, we have to find the appropriate terms, how to avoid some kind of a misunderstanding or confusion of others. So that's some issue. I'm always also headache with this kind of terms, Always our scientific way, we need a standardized protocol. We need a standardized sampling strategy. So what kind of standard is standardized? Something. So, right, I think we try to generate the problem by ourselves.
I think that can be discussed in the general discussion. So maybe Christian?
So I don't think I can give you a volume that would be a one-fits-all. But what I could say is that the standards can have a passage where it says that you must specify the volume that you used, so that you later on then can look at the data and you know exactly how much water was filtered, so that you can assess the reliability of that in hindsight. So it will not give you the perfect result for your analysis, but it will future-proof the data for later backwards comparability. So you can check whether that's okay. And if you find that your data is consistently underperforming, then you need to change the standard and define a volume that is higher than something.
That's
a cutoff.
That's an adaptive approach.
Okay. I have a very quick question from the online. So, are there any plans to expand the standardization for other types of samples, such as sediment? at this moment?
At this moment, we do not have the plan to standardize the method other than water samples, but we should do.